Hello and welcome everybody. Today I will be presenting our work where we combined conditioning generative adversarial networks together with phenotypic image features to guide the novel design of small molecules. And although I will be presenting all the slides, at the end of the presentation, both Oscar, Mendes Lucio, and me will be available for questions and for further discussion. So starting with the background and the motivation, so it is well known that the design of new molecules with a desired biological activity is an extremely difficult task. And there have been several computational approaches proposed to tackle this kind of problems. And actually recently we published one of such approaches in which we propose to combine conditional generative adversarial networks, so CGANs, together with gene expression profiles in order to guide the generation of small molecules. And to understand a bit the motivation for this framework, we should start then with a small recap of what CGANs are. And this type of models consist then of a generator network which samples Gaussian noise, so which generates given samples in these example images by sampling a Gaussian noise, and a discriminator network who is in charge of classifying if an image or a sample was generated or if it was a real sample. And these two networks are trained in an adversarial manner, so competing against each other such that they improve each other during the training process. And the conditional variation of these GANs consists of a small modification where we feed both the generator and the condition and the discriminator with a condition vector in order to guide or to give the model more control on which type of sample to generate. So in this case, if we feed a condition, so as a condition generate the number 8, the generator will take that into account. So we'll take the condition into account and generate the desired class. And then now the discriminator, which should also have the task of classifying real versus generated, but also so deciding if the condition matches the generated sample or not. And there are different types of architectures that you can choose here. You could choose to have the condition and the discriminator, so the condition classification and the discriminator in one network, but we chose to have them in different networks. So we have a condition network which classifies match versus not match, and the discriminator which classifies real versus generated. All right, and then our idea was then to use this framework to generate small molecules instead of digit images, and instead of using integers for the condition, then conditioning the generation process on gene expression profiles. And with this, our final goal was then to train a model which could generate small molecules which had a high probability of inducing a given gene expression profile. However, everyone knows that gene expression is not so cost effective, and therefore in this project or in this presentation, we wanted to evaluate if cell painting would be an alternative approach to these gene expression profiles. Okay, so going a bit now more into the architecture of the model and the details of the training. So this is the basic model that I showed before. One detail is that we don't feed directly the images to the generator network, but we use Cell Profiler to calculate morphological profiles, and we feed those profiles as the condition to the generator and the condition network. And the second detail is that our generator doesn't generate directly chemical structures, so it doesn't generate chemical formulas, but it rather maps the Gaussian noise to a molecular embedding space, and then we use a decoder network to decode this embedding into a chemical formula. So to conclude, the model consists of 4 different models, which are trained in different data sets and at different stages, and I will go through each of them now. The first one is the decoder. So the decoder consists of a variational autoencoder, which is trained to translate different representations or equivalent representations of the same molecule. And since these molecular representations are sequence of characters, then the variational autoencoder consists of recurrent neural networks or with gate recurrent units. And this decoder is then trained on 1.25 million molecules extracted from ChemBall22. Then going to the condition network, the condition network is trained on the BBBC36 dataset, which contains morphological profiles for approximately 30,000 compounds. And the architecture of this network follows a very simple, fully connected architecture. The molecular embedding is processed by a 2-layer network. The morphological profiles is processed by a 3-layer network. Then these two networks are concatenated, fed into a further layer, and then classified by a sigmoid function between match and not match. Another detail is that this condition network is trained, is pre-trained before the adversarial training, and the weights are frozen during the training of the generator and the discriminator. And finally, going to the architectures of the generator and the discriminator, they follow very similar design principles as the condition network. The Gaussian noise is processed by 2 fully connected layers, the cell profiler features by 3 fully connected layers, followed by concatenation, another layer, and finally a tan-H activation function to generate the molecular embedding. And the discriminator just follows a 3 fully connected classification network to classify real versus generated samples. And this, again, these two, generator and discriminator, are trained on the BBC36 with 30,000 compounds. Okay, and before going to the results, I first want to walk you through which evaluation metrics we used to evaluate the model. And as you will see later, how we evaluate the model is always by comparing the similarity distributions between different sets of molecules. So the idea is that we pick a set of conditions or a condition group. So this could be, for example, all the profiles belonging to a specific compound. Then we generate 20,000 molecules using those conditions. And then we compare the similarity of those 20,000 of this set of 20,000 molecules against other sets. And these other sets could be molecules generated with a different condition, but it could also be annotated data sets, so external data sets. How we compute the similarity between the different sets is that we take for every molecule in the first set, in the left set, we find the closest nearest neighbor in the second set. For here we're just showing 2, but we compute up to 10 nearest neighbors. And we compute the similarity between the 2 closest neighbors, so sim1 and sim2. And we do the same for the next molecule and for all the molecules in the reference set, so the left set. And at the end, we collect all these pairwise similarity values and then product the distribution of the similarity between the different sets. And finally, so how we compute similarities between 2 molecules, so these pairwise similarities here. For this, we use the dye similarity on ECFP fingerprints. So this is basically a very standard measure of similarity between compounds, which roughly reports the proportion of shared molecular substructures between the 2 compounds. And now diving into the results. The first thing that we examined was the relationships between morphology and chemical structure. So basically the question that we wanted to answer here was whether the model can translate morphological similarity into chemical similarity. And to address this question, we clustered the morphological profiles in the training set using k-means clustering on the per compound median profile. And after clustering, we used the cluster centroids to build a minimum spanning tree, and in order to recapitulate the hierarchy of the clusters and to get a feeling of their organization and the distances between different clusters. And also to have a reference point, we we named cluster 0 as the cluster with the most DMSO samples, and we numbered all the other close clusters by increasing distance to this DMSO cluster. And once the clusters were defined, then we generated 20,000 molecules conditioning on profiles from each of the clusters, and then compared the similarity distribution of generated molecules against those from cluster 0. So here we show the results of comparing the similarity distribution between molecules generated on profiles from cluster 2 versus profiles of the DMSO cluster. And what we can see is that if we move further away from cluster 0, so remember the clusters are numbered by increasing distance to the cluster 0. So if we now check the similarities for cluster 8, we see that we move to lower similarities. If we go further to the left, cluster 15 also reduces the similarity, and finally cluster 18 shifts it even more to the left. So these results assure or show that our model was able to translate morphological similarity into chemical similarity. Okay, and then the next thing that we wanted to check was, that we aimed to do, was to compare the generated molecules against external data. So the question we wanted to answer here is how close can we get to annotated molecules from external data sets, so molecules that were not included in the training set. And our idea with this would be to take a group of conditions. Let's say we will take all the profiles from CDK inhibitors, generate 20,000 molecules based on those conditions, and then compare the similarity distribution of those generated molecules against known CDK inhibitors that were not in the training set. So that means then that we will need to fetch these inhibitors from an external data set in order to perform the comparison. We tried to do this with mode of action annotations because many of the compounds in the training set had annotated mode of actions, but we found that the annotations were very noisy and that we couldn't really make much sense of the results. Therefore, we then decided to use profiles or use conditions from a different data set, which was the BBBC 37 dataset, which has which reports overexpression profiles for 160 genes. Of course, we made sure to process the profiles from this dataset exactly in the same way as the profiles from the training set, and we visually compared the profiles, and they didn't seem to follow very different distributions. And as an external data set to compare against, since we know that overexpression should be an activating perturbation, then we searched the ESCAPE database for agonist compounds against the genes in the BBC 37 dataset. And then we compared the two datasets. So doing this, we found agonists for 6 of the 160 overexpression genes. For some of them, we had a lot of samples, and for some of them, we only have 4 agonists. Okay, and besides conditioning on these 6 genes for our experiments, we also included a set of reference genes and DMSO, which we picked as the top 10 distinguishable genes, and those genes were included also in the experiments just as a reference. And finally, the last thing that we want to point out is that for this analysis, we didn't examine the full distribution of similarities between the generated molecules and the ESCAPE agonists, because we found that they were these distributions depended a lot on the set of compounds, on the set of agonists that we had here. So instead of looking at the full distribution, we only focus on the tail of the distributions, which means we only focus on those agonists for which we could generate molecules with a similarity higher than 0.8. And a 0.8 similarity threshold is a very strong threshold, so that means that we're getting very close to the agonists. And to get some statistical power, we repeated the generation process 3 times using different random seeds, and we detected significant hits by comparing the counts against the DMSO controls. All right. And the first result was that we couldn't generate close enough molecules for neither HSP-A5, STAT3, and JONC. So we could never get higher than a 0.8 similarity threshold, but we could generate some reasonable results for the other 3 genes, which I will present in the next slides. So starting with TP53, so here we're comparing generated molecules against TP53 agonists from the ESCAPE database. So basically what we're trying to plot here is how many times do we get close to a TP53 agonist when conditioning on TP53 overexpression profiles or on DMSO and the top 10 genes. And remember that DMSO and the top 10 genes are just there for a reference to check how specific the conditioning works. And here we're plotting, so each plot, one plot per ESCAPE agonist. The x-axis corresponds to the condition gene, the gene that was used to as a condition to generate the molecules, and the y-axis is reporting the number of compounds with similarity higher than 0.8 to that specific ESCAPE agonist. And the first result that we can see here is that we could get significantly close to 7 TP53 agonists out of the 12,000 agonists that we had available. And we can also see that 4 of those hits occurred when conditioning on TP53 itself. So those are what I call TP53 specific hits here on the left. And however, we also got 3 of those hits occurring on different genes than TP53. So I call them on a specific hits here on the right, because that means that we get very close to a TP53 agonist when conditioning on a different gene than TP53. And okay, but even though we get some on specific hits, we still can see that the majority of matches to TP53 agonists happens when we condition exclusively on TP53. And also the most strongest or significant results happens when conditioning on TP53. So this is rather encouraging. Another thing that we can notice from this data set is that all the TP53 conditioned agonists look very similar to each other. And this is just showing that the model found a chemical pattern which was associated to TP53 profiles. We were also interested in knowing how close the agonists were and the generated molecules were to those molecules in the training set. And here in the second row, I'm plotting the ESCAPE agonists, so for TP53. And in the third row, I plot the closest molecule in the training set. And as you can see, indeed, we had for all the 4 agonists, but also for the unspecific hits, we have very similar molecules in the training set to the ESCAPE agonist. So thinking about it, this is not necessarily a bad thing. This just shows that the model was able to learn relationships between phenotype and chemical structure in the training set, and then the model is doing a good job interpolating between molecules in the training set. Okay, and then here I show the results when comparing against NF-kappa-B1 agonists. So again, what we're trying to check is how many times we get close to a NF-kappa-beta-1 agonist when conditioning on NF-kappa-beta overexpression or these other referent genes that we have there, including DMSO. And in this case, we could only get significantly close to one of the ESCAPE agonists out of 185. And this hit happened only when conditioning on NF-kappa-beta-1. Here again, I show the ESCAPE molecule and the closest molecule in the training set. And we can see that we again had a very similar molecule in the training set. And if you look at the generated molecules, you can clearly see how the molecule is how the model is interpolating between chemical structures. All right, and finally, we report the results for the last agonist, which was BRCA1. And in this case, we could get significantly close to 18 BRCA agonists out of the nearly 8,000. So again, those specific hits, those were all the hits, sorry. And from those 18 hits, out of 8 hits occur when conditioning on BRCA itself, so the specific hits on the left, and 10 of those hits happens when conditioning on other genes. And so even though we have rather some on specific hits, we still see this as a positive result because the largest proportion of matches against BRCA agonists happens when we condition on BRCA overexpression profiles. And indeed, we didn't follow up on this on specific hits, but there can be many reasons for this. So one of the reasons is that BRCA, for example, doesn't have a very strong phenotype compared to the other genes. It could also be that the phenotype of BRCA is very close to some of the specific hits reported here, but it could also be that these escape molecules reported here have polypharmacology and could hit several targets at the same time. So it's just a lot of speculation, and we didn't follow up thoroughly on these other hits. But still, the larger proportion is generated when conditioning on BRCA, and those are still very encouraging results. All right, and the final thing that we wanted to check was we were wondering if we would get more impermeable compounds when we condition on DMSO profiles. And the reasoning behind this is that DMSO, so this neutral phenotype, should not only include inactive compounds, but it should also include compounds that couldn't pass the cell membrane, and therefore couldn't have an effect. And to check this, we run the generated molecules on a in-house permeability prediction model, and then and plotted the distribution between generated molecules conditioned on the top 10 genes versus DMSO profiles. And from the distributions, we can see that by conditioning on the top 10 genes, we move slightly towards higher permeabilities compared to DMSO. However, these results are not significant. Okay, and finally, now coming to the conclusions. So the main conclusion is that our approach provides a proof of concept for the applicability of morphological information and morphological profiles to the design of new targeted small molecules. And for example, we have shown that the model can learn relationships between morphology and chemical structure. We also showed that conditioning molecule generation on morphological profiles can produce similar compounds to known actives with reasonable specificity. And finally, another aspect that we think is worth pointing out is that the model showed that it is possible to condition not only on chemical profiles but also on other type of perturbations such as gene overexpression, and that makes it very flexible, and it opens a set of possibilities to condition. When you have an unknown target, you could condition on the profile of the knockout of this target, so for a novel target, and then try to check if the model can generate molecules that reproduce that phenotypic profile. And looking at the prospective work or outlook, so we think that to push this further, we will need to apply this to a larger chemical space. And we hope that using a larger chemical spaces will help generalizing to molecules outside the training set. So not only interpolating inside the training set, but also learning chemical patterns of activity and extrapolating. And also that by having a larger training set, we hopefully could then train also the model end-to-end, in which we mean also include the feature extraction during the adversarial network process and skipping then the cell profiler profiles calculation. All right, and